In spite of all their difficulties, people with Rett have attractive qualities which make it rewarding to work with them. Their interest in everything and everyone is genuine, and real relationships can be developed. They enjoy acquiring and using skills of which they are capable, and they can be persistent in learning. Their enjoyment of human company is endearing, and when free from frustration or discomfort, they are happy people. Spontaneous movements are generally much better than planned movements, and the skills of people with Rett are such that they can enjoy shared activities. They can benefit from riding and swimming or moving freely in water. Vision and hearing are usually good, and these senses, protected and valued, provide avenues for shared pleasure and for learning. Appreciation of music is a very special asset for people with Rett disorder. Music is a major source of pleasure, and in addition provides a way to interact with other people, to take turns, express emotions, and develop hand and vocal skills. People with Rett demonstrate individual choice and can be clearly moved emotionally by music, soothed, stimulated, or simply satisfied. This musical sensitivity deserves careful research and offers a channel for practical therapeutic intervention. What we have shown here of the nature of the Rett disorder indicates many possibilities for therapies to support people throughout a happy and healthy life. Now, in 2005, we appear to be on the brink of discoveries which it is hoped will lead to effective pharmacological and genetic intervention. Dr. Adrian Bird is leading research at Edinburgh University. We asked him what intervention might in future be possible if the diagnosis is made early in life.
Well, I suppose the obvious intervention would be gene therapy, because these individuals are lacking in at least a fraction of their cells, functional MECP2, MECP2, and one could, if gene therapy was a reliable, into the brain, were a reliable technology, one might be able to express the gene. And there are, in fact, people trying this via, the methods are quite crude. Gene therapy itself is not a widely used technology, but the advantage of this is you wouldn't need to understand too much about what MECP2 was doing. You just simply put it back and let things happen. There are risks because it's now known that if you express this protein at the wrong level, that can be bad. So under its own normal promoter in its proper gene context, it works at the right level, but introduced gene may not. So that's one obvious possibility, but is quite a way off, I think, to be therapeutically useful. The frustrating thing about Rett syndrome, I suppose, is that every cell that is causing this disorder has both a non-functional and a functional MECP2 gene. And so the other thing one would really like to be able to do is simply switch on the functional MECP2 gene that has been silenced in half of those cells. That is easy to say, not so easy to do. But potentially, if one understood exactly what it is that's holding that gene off on the inactive X, because they're expressing the wrong gene in those cells, one might be able to dig it out, and that would be a pharmacological approach. And the other way is to study the genes that are misexpressed as a result of the absence of MECP2. The assumption might be that they're going to be overexpressed because it is a repressor. And if they're overexpressed, perhaps one could use a drug that inhibited them to take out the worst offenders as far as causing this phenotype. Just to come back to what you initially said, if one were to pick it up early enough, could one do these things? It is not a foregone conclusion yet that once the symptoms have arisen, that they could be reversed. And that's something that I think people are very interested in checking in the mouse. After the symptoms have already arisen, can you administer the protein and make things go backwards? because, as you well know, this is not a neurodegenerative disorder. The neurons do not die, they remain in place, and so the extent to which they could be rescued by re-expression of the protein is an untested possibility. So in the best of all possible worlds, you could take some with the symptoms, administer the gene in some controlled manner, and then watch the symptoms dissipate. But that is, you know, a dream yet to come true.
Professor Michael Johnson, a neurologist, is carrying out research at the Johns Hopkins University in Baltimore.
Mike, what do you think that MECP2 is actually doing at the synapse in Rett?
Well, I think that it's an important break for control of substances like brain-derived neurotrophic factor that are supposed to respond to patterned activity at synapses. And when that break is lost, then the sharpness of the synaptic formation is lost. In other words, the signal, when trophic factors are allowed to go to too high a level, then it reduces the signal to noise that's necessary for close establishment of synapses.
So when do you feel that that has its impact on the developing brain?
Well, the greatest impact clinically is in that postnatal period, and I think that the impact may be greatest in neurons that experience this great proliferation of synapses and then pruning, such as the cerebral cortex. And this seems to be important for higher cortical functions like speech and fine motor control. So I wonder if BDNF isn't disproportionately more important for circuits like that in the cortex, and therefore its effect is felt in the postnatal period more than in the earlier period.
And what future do you see for intervention in Rett?
Well, I think if we could intervene at the genetic level, that would be good, but I think also in analogy to disorders like PKU and other genetic metabolic disorders, that there may be a way to alter synaptic formation with drugs that act at synapses, analogous to Parkinson's disease and Alzheimer's disease. There may be ways that aren't harmful to modify synaptic activity that could improve the overall outcome.